Today we will discuss about some of the anti-cancer strategies that can be used against platelet tumor interactions. As we have discussed in our previous videos, uh, like a couple of previous videos, that how the platelet tumor interactions helps in the cancer migration and metastasis. In this video, we will understand how we can target those platelet tumor interactions to reduce the cancer migrations, metastasis, and growth. So these strategies are being divided into two different categories. First one is generating. First one is about the antibodies that can inhibit the interactions between platelets and the tumor cells. So there are three different antibodies are being discussed. First, antibodies that can target GP6 or glycoprotein 6 that are expressed on the platelets, and the second antibodies that can target CLEC2 that is also expressed on the tumors, and third one is this integrin alpha 6 beta 1 that is also expressed on the platelets. So if these antibodies can interact with the platelets cell surface proteins, and thereby preventing its interactions to its counterpart that are present in the tumor cells. For example, GP6 interact with this galactin 3, CLEC2 interact with this podoplerin that we have discussed how it will trigger the intercellular signaling pathways in the tumor cells in our previous videos. And this integrin alpha 6 beta 1 prevents its antibodies against integrin alpha 6 beta 1 prevents its interaction with this ADAM9. That comes from the family of matrix metalloprotease 9. And apart from targeting those cell surface protein that are expressed on the tumor cells, one more things can be used, that is in the activated platelets there is upregulation of this PITP alpha and PKC alpha that will leads to the high production of this or high expression of this surface PS. And this high expression of the surface PS is not necessary for maintaining the homeostasis. That is one of the role or critical role of the platelets. So one can easily target this signaling pathways such as PITP alpha and PKC alpha. So small potential molecules can be used, or we can say potential for small molecule inhibition. Yeah, small molecule inhibitions can be used to target specifically to this signaling pathways to reduce the high surface PS formations, and that also helps in this tumor cells migration and metastasis. So by targeting these two points or these two strategies on the platelet cells, one can reduce the interaction of the tumor cells with the platelets and helps in the detection of the CTC, that is circulating tumor cells, by the immune system. The next approach is about this modifying platelets. So there are different approaches can be used. First, this membrane can be purified from the platelets of either of the patients that are affected with cancer, or from the other patients or animals, and then that membrane can be loaded with the nanoparticles, as the platelets has the natural tendency to be attracted towards tumor cells. This membrane coated with drug coated nanoparticles on the membrane of the platelets can be used to target the tumor cells, or decrease the efficiency of the tumor cells. So this is one of the process that in which the platelets membrane purifications can be done from the platelets. And the next way that these platelets can be used is this detergent extraction. From this, the platelet decoy can be extracted, and which is devoid of any other platelets content, and then this decoy can be modified in a way that can target the tumor cells, or that can kill the tumor cells. And the other things is that the platelets can be engineered with the antibodies that can generate apoptosis or other uh, tumor suppressing properties, or the antibodies that can have more another tumor suppressing properties. Next one is this platelet activations. The platelets can be activated that contains the platelets that can be activated in vitro or in the lab, and thereafter the exosomes that are released by the platelets, and these exosomes can be modified in a way that it contains the drug, and these drug loaded exosomes can then be used into the patients that are affected with cancer cells, where these drugs can help in suppressing the tumors in the patients. So these are some of the ways by which these anti-cancer strategies can be used to target platelet tumor interactions, to reduce the tumors, or to diagnose the patients that are affected with the cancers. So that's all for this video. Thank you for your time.